Hi, I'm Christy Burton. I'm a research associate working at the Hospital for Sick Children. I'm going to talk to you today about ADHD, what it is, how we measure it, and what genes have to do with it. So let's start with what is ADHD. So I think a lot of you probably know that ADHD is a psychiatric disorder that's characterized by inattention, hyperactivity, and impulsivity. And these symptoms need to be developmentally inappropriate. So they're worse than their same age peers. They also have to be impairing, and we want to see them in more than one setting. So for example, both at home and at school. Based on the DSM, which is our diagnostic manual or Bible here in North America, there are three possible presentations for ADHD: predominantly inattentive, predominantly hyperactive impulsive, and combined, where you meet criteria for both of the subtypes. And this presentation is actually the most common for ADHD. Cognitive deficits are very common with ADHD, so things like difficulties with response inhibition and working memory, a lot of executive functions, but they're not always present, so they're not part of the diagnostic criteria. So a little bit of a primer on the epidemiology of ADHD. So ADHD has very much been thought of as a childhood disorder, although we know more now about ADHD in adulthood. So the prevalence in kids is about 5.3%. It's actually one of the more prevalent psychiatric disorders in childhood. And then that prevalence goes down to 2.5% in adults. And we do know that hyperactivity symptoms do tend to wane with age. In childhood, it's much more common in boys. So we see a 4-to-1 sex ratio of boys to girls diagnosed with ADHD. And then in adulthood, it evens out about 1-to-1 ratio for men and women. And ADHD is very comorbid with many disorders. So in childhood, things like autism, learning disorders, externalizing disorders like oppositional defiant disorder, but also a lot of speech and language problems are very common. So reading disorder, language impairment, and a lot of pragmatic language difficulties. Mood and anxiety is prevalent with ADHD across the lifespan. And in adulthood, we start to see an increase in comorbidity of substance use and personality disorders, which obviously are not diagnosed in children. So next question is, how do we measure ADHD? And before I get into the actual measurement scales that we use, I want us to think a little bit about what is the goal of what we're measuring, or why we're measuring what we're measuring. And so usually in a clinical setting, we're trying to establish if people have a diagnosis or they don't. In research, we often want to group people into cases or patients or controls. And this approach is very useful and serves a lot of purposes, but I want us to think about how that may not be capturing the whole story. So think about yourself, think about people you know, and think about inattention, how inattentive or attentive people are. Now think, do people tend to fall into two groups, people that are inattentive and people that aren't? Or is it that people kind of fall on a spectrum? So some people are super inattentive, some people are kind of inattentive, some people are just normal, and some people are super laser focused. So my guess is that you're probably going to sort of break people out onto a spectrum. And we think a lot of disorders are composed of traits that function very much this way, that they're distributed in the general population, kind of like this. This is going to apply to like mood, anxiety, obsessions, compulsions. And I think that's a big part of the reason why at the beginning of all psychopathology textbooks, there's always a warning, like don't diagnose yourself and people you know. And I think it's so easy to fall into that trap because there's bits and pieces of those disorders that seem familiar to us and people we know in ourselves. Doesn't mean we necessarily have those disorders, but it's just that these traits don't exist in an all or nothing space. So we think that something like ADHD is going to be composed of these traits, where there's some people that are super inattentive, hyperactive, impulsive, and then there's going to be a gradation across the spectrum with a whole bunch of people sitting in the average position in the middle. And then people that actually have a diagnosis of ADHD are going to be at the high end of that spectrum. It's not always going to work out exactly that perfectly, but that's generally the idea that we have. So now that we have that sense of ADHD as being a quantitative trait, the question is, how do we measure it? And how do our measures, do they accurately capture that? So one example of a typical ADHD measure, so ADHD is diagnosed with a clinical interview, but there's a lot of rating scales that are used both clinically and in research. And one example is the Commerce Rating Scale. So this is very typical of most ADHD and a lot of psychopathology scales just across the board. So you'll have a symptom, let's say it's inattentive, and then you're going to rate that child from not having that symptom up to some positive integer. So they have it a lot, like a score from 0 to 3. Now, this distribution is going to look a little bit different in an ADHD clinic. You're going to see a lot more people at 3, but it may look like this in a general psychiatry clinic where not everybody has ADHD, or in the general population, which is a population that I work with quite a bit. So the question is, who are these people at 0? Are they normal? Are they hyperattentive? You can't really pull it out when you have this clustering of people at 0. And so ideally you would like a scale that measures, sort of pulls apart the variation and separates people into more fine-grained categories, for lack of a better word. So this is sort of the principle of the strengths and weaknesses approach that's used in the SWAN, but other scales as well. So the SWAN is like other ADHD scales in that it queries the 18 symptoms that are found for ADHD in the DSM, our diagnostic Bible, and it allows us to break up scores into the total overall ADHD scores and the inattentive and the hyperactive impulsive subscales as well. But it's framed very differently. So instead of asking about inattention or hyperactivity, it frames it in terms of the strength. So instead of inattention, can you sustain your attention? And then you're able to rate the child from average, which is a score of 0, to far above their peers or far below their peers. And so you're able to see a more normal type distribution in theory. Now, one thing I want to point out about the SWAN is that whenever we work with it, the data, we always reverse the scores. So high scores mean, high SWAN scores mean high ADHD traits. And hopefully that'll be clear when I show you some of the data from this measure. And if you want to access the SWAN, you can do so on our website, and you can use the link here below. Now, what is still unclear, so now we're pulling apart that 0 and we have this whole strength end of the distribution. But are they strengths, or is it maybe another psychopathology? So if you're really hyper-focused or inactive or very, very careful about your decision-making, is that a strength necessarily, or is that potentially the sign of another psychopathology like OCD or anxiety? And the only way to find out is to test them. Now I want to talk about what genes have to do with ADHD, and also how the SWAN might help us understand the genetics of ADHD a bit better. So we know that ADHD runs in families, and you don't need to be able to understand this figure exactly. So these are pedigrees. Everyone's a different family. Every symbol represents a person, and each row is a generation in a family. And the black and gray symbols represent people with ADHD or with subclinical ADHD. And I think what's very clear is that ADHD is clustering or running within families. Now, that does not mean necessarily on its own that ADHD is genetic. There could be other factors that are contributing to that. But what we can do is look at twin studies. So we can compare identical or monozygotic twins to fraternal or dizygotic twins, and that allows us to disentangle the relative contribution of genetics and environment. And what we know from many, many ADHD twin studies is that genetics play a really big role. So about 3/4 of the variance is accounted for by genetic factors compared to 24 for the environment. And actually, ADHD is one of the more heritable or genetic psychiatric disorders. Now, we also know that there is no ADHD gene. There's no one gene that's causing ADHD, just like there's no good at basketball gene or tall gene. A lot of psychiatric and medical disorders and a lot of traits like height, they are polygenic, which means that there are a lot of genetic variants that contribute to their risk. And when I say a lot, I mean like thousands to hundreds of thousands of genetic variants involved. And that's what I'm hoping to illustrate here in this figure. So this comes from the largest ADHD genetic study that's been published to date from the Psychiatric Genomics Consortium. And all you need to know is that each band of color along the x-axis, those are chromosomes. And then each dot represents a genetic variant or a SNP. And to give you a frame of reference, so if you think of the genetic code as being a book and there's 3 billion letters in it, each dot represents 1 letter. And so actually on this figure, there's over 10 million letters or SNPs represented. You just can't pull them all apart because there's so many. So right now for ADHD, we know there's at least 12 replicated genetic variants that we're confident about, but we're only just starting to scratch the surface. It takes very large samples to understand the genetics of a condition. And so as we get larger and larger samples and we get better statistical power, we anticipate all those peaks to rise. And then all these variants that are below the red line are actually going to be contributing risk to ADHD. And so what we think is going to happen for, and what we think is the case for a lot of common disorders, is that genetic risk is going to be normally distributed. So there's going to be a small proportion of people that have high risk for the disorder, a whole bunch of people that are average, and some that are low. Nice Gaussian distribution. And I put this figure up so you can remember the distribution I showed you for the SWAN, and that's very much what we're hoping. So the SWAN is going to capture the full spectrum of variation in terms of ADHD symptoms and behaviors, but it's also going to capture the whole spectrum of ADHD genetic risk. At least that's the hope. And so the SWAN scale is relatively new, newer than a lot of the other ADHD scales that are available. And so part of the process that we had to undergo is everything I've presented to you is what we think, our rationale for the design of the SWAN, but is that actually true? Is it psychometrically valid? Is it actually measuring ADHD? And is it going to be useful for genetic work? And that's the basis of this paper that we published last year in JCPP. If you want to access the paper, you can do so at this link. So there was a couple questions we wanted to ask, and these are general questions that you would want to ask about a new scale and the type of information you want to have about scales that you're using both clinically and in research. So is the SWAN similar to other ADHD scales? Does it have good convergent validity? Is SWAN associated with, or can it predict ADHD diagnosis? We want to make sure that the SWAN is measuring traits and that we think ADHD is at the high end, so they should be strongly associated. What do the strengths on the SWAN represent? We need to know if we're measuring just low ADHD traits or if we're actually picking up on another disorder like OCD or anxiety. Excuse me, and also is SWAN associated with genetic risk for ADHD? In other words, is this going to be a useful scale for us to understand the genetics of ADHD? So this study used 3 different groups of samples. So most of the work was done in a community sample, Spit for Science, which our group created and runs. So these are kids that were recruited at the Ontario Science Centre. We just started Spit for Science 2, but the original Spit for Science 1 had over 15,000 kids, aged 6 to 17, and we measured a whole bunch of different behavioral and cognitive traits, but the ones I'm going to talk to you about today is ADHD traits were measured with the SWAN, OCD traits were measured with the Toronto Obsessive Compulsive Scale, which we also designed, and anxiety traits using the Child Behavior Checklist or the CBCL. And then we collected genome-wide data from their saliva, which is why it was called Spit for Science. And then to validate a lot of the work that we did in the community sample, we used the ADHD clinic sample at Sick Kids, which they were recruited by Drs. Russell Shacker and Jennifer Crosby, who are also the PIs on Spit for Science. And so we had a sample of kids with diagnosed ADHD and controls, same age range as the community sample, and the diagnosis was made using standard clinical diagnostic assessment by a psychiatrist or psychologist. And then we also measured ADHD traits using SWAN so we could compare it to what we saw in the community sample. And then for our genetic risk, we took data from the Psychiatric Genomics Consortium, which is one of the largest consortia in the world. And they have data on genetic risk for ADHD, OCD, and anxiety. So we use that data. So the first thing I wanted to show you before I'd shown you sort of that like graphic of a distribution of how we thought ADHD traits were going to be distributed, which was the hypothesis. And I'm showing this to you. So this is actually how ADHD traits are distributed in our Spit for Science sample, which is a general population sample. So we do see that clustering of scores at 0. So a lot of kids are average, but then you have people at both ends having smaller number of people with high ADHD traits or low ADHD traits. So now getting to our questions. So first, is SWAN similar to other ADHD scales? So we're looking at convergent validity using Spearman's Rho, which is essentially correlation between ordinal factors, and we compared it against the Conners rating scale, which is a very established measure of ADHD. Now, one thing I haven't talked to you about yet is the source of information. So in Spit for Science, we collected information from parents about their children and then from teens about themselves. So parent report or self-report. And the source of information for ADHD, but for a lot of psychiatric disorders, is very important. So we know that for ADHD and other externalizing disorders, parents tend to be better raters of their children's behavior, or more accurate raters of their children's behavior, than teens are of themselves. They just have less insight into their own behavior. It's actually the opposite for internalizing disorders, where kids have a better sense of what's going on in their head, and maybe their parents aren't aware of how anxious or depressed they are. So when we look at the Spearman's Row, it's actually very high for parent report, which is great. Self-report, moderate, so not as good. But again, not surprising based on what we know about parent versus self-report for ADHD. Second question is ADHD associated with, or can it predict, an ADHD diagnosis? So first, we just want to see what SWAN scores look like in people with a diagnosis of ADHD, which are the darker blue circles, compared to people without a diagnosis with ADHD, which are the light blue circles. And what we see either in the community sample or in the clinic sample, same effect. SWAN scores are significantly higher in patients with ADHD versus controls. It doesn't matter if you look at the total score or the two subscale scores. Very consistent, which is a great start. Second question is, are we able to predict ADHD both sensitively and specifically, or with specificity in a clinic sample? So creating cutoffs using an area under the curve analysis, and then testing those cutoffs in a clinic sample where we have established clinician diagnosed ADHD. And what we see here is that generally sensitivity and specificity are pretty good. The one place where it falls down a little is SWAN self-report. Again, for the same reasons that I just explained. And what we see is that SWAN self-report is more likely to have false negatives. So essentially, SWAN is more likely to not identify individuals or teens with ADHD that a clinical interview would identify as a case. And again, not super surprising, but these numbers aren't terrible. And what do the strengths on the SWAN represent? So in this case, remember I was saying to you that we think that potentially those low, the strength end of the distribution might be representing another disorder. So we divided our community group by their SWAN score into low, medium, and high. So low, medium, and high. And then if we think, if the low SWAN scores are actually representing OCD or anxiety, then we would expect the people in the low group to have very high OCD or anxiety trait scores. And that's not at all what we see. 0 is a very average score for these standardized scores. So essentially, there's actually no association with OCD traits and SWAN. And for anxiety, we do see an increase in anxiety traits in high SWAN scores. And again, that's not super surprising. As I told you before, anxiety is a very common comorbidity of ADHD. So it's not surprising that we've seen an elevation of anxiety traits in a high SWAN group. And our last question is SWAN associated with genetic risk for ADHD? So in these analyses, what we do is we take genetic risk from the PGC, those really large genetic samples, and then we see if that is associated with SWAN scores. So essentially, if the SWAN is picking up on ADHD genetic risk, what we want to see is that a high genetic risk for ADHD from the PGC is associated with high SWAN scores. And that was exactly what we saw. So it's not super important to understand what all the colors mean here, but what I do want to point out to you is that these bars actually represent thousands of genetic variants, just reinforcing the idea that there are many genetic variants contributing risk to ADHD. And getting back again a little bit to the strengths question, you know, is this measure picking up on ADHD genetic risk specifically? Or is it tapping into general psychopathology, or is that low end picking up on another disorder? And that's not at all what we saw. So genetic risk for OCD or anxiety was not associated with SWAN. So essentially what we're seeing here is that the SWAN is specifically picking up on ADHD genetic risk, but not risk for at least these two disorders. So just to quickly summarize what we found, getting back to the question, so is SWAN similar to other ADHD scales? Yes, it has good convergent validity with an established scale, although it's better for parent versus self-report. Is SWAN associated with or can it predict ADHD diagnosis? And again, the answer is yes. And again, better for parent than self. And what do strengths on the SWAN represent? I think we can say pretty clearly that it's probably not OCD or anxiety. But we can't rule out other disorders. And obviously this, like everything in genetics, this result needs to be replicated. And is SWAN associated with genetic risk for ADHD? Definitely yes, and it does seem to be quite specific for ADHD. But just like for the previous question, we need to make sure that it testing out a larger range of genetic disorders to make sure that it's actually specific. And so if you don't remember anything else about my talk, what I do want to remember is a couple things. So ADHD is a complex childhood onset common disorder. ADHD represents the extreme of traits that are seen in the general population. It's really important to consider the full spectrum of traits. So often in clinical and our research, we really focus on the high end, like the weaknesses side, but I think there's still a lot of information that can be gained from the strength side for both clinical and research applications. And it also often provides you with a lot more power than a typical case control design when we're thinking about research. SWAN is psychometrically valid and useful for ADHD research, both phenotypic and genetic. It's important to consider the reporter of information. Parents are going to be better reporters for some things, and kids are going to be better reporters about themselves for others. And it's really important to understand what those differences are and consider them when you're using different scales. And we can't assume what our rating scales measure. So obviously we know it's really important to be validating our scales against existing scales and making sure it's picking up on the construct that we think it is. But it's equally important to make sure your scale is not picking up on something else that you didn't intend it to, and also be mindful of those properties and scales that you're using. ADHD is genetic, and there are many, many genes that are going to be involved. There is no ADHD gene. It's very polygenic. And for that reason, and I didn't really talk about this much in my talk, but I wanted to bring it up because it comes up a lot whenever we talk about genetic risk scores, is there is no genetic test for ADHD. So the genetic scores that I showed you, those are very useful in terms of research. They're being used extensively, and they can tell us a lot about whether two disorders or traits share genetic risk. But they're not ready for use in clinical settings yet. And we do anticipate that at some point they will be useful to help identify potential diagnostic subgroups or sensitivity to medication or potential risk for another disorder. But we don't think that a genetic test is ever going to replace a clinical diagnosis or assessment of ADHD. It may just be used to complement it. And so that's it for me. If you're interested to find out more about the research that we do or more about the paper, you can find them at these links here. I'm really sorry I couldn't be there for the question and answer period, but please feel free to email me. My email is below on the slide. And yes, thank you for listening and best of luck with your course.